Today's lesson is about cellular transport. It comes from chapter 2, section 3 of your science textbook. And before we get started, we're just going to go over the lesson objectives. Today we want you to be able to explain how the processes of diffusion, passive transport, active transport happen, and why they are important to cells. We also want you to be able to explain and predict what's going to happen if the solution is hypotonic, hypertonic, or isotonic on a cell. And so we're going to set up your notes. The first thing that you're going to do is set up some Cornell notes that are going to look like this. So on your paper at the top, put cellular transport. Down the left-hand side is where you're going to put your terms. I've started and listed a few there for you. And then make sure you leave some room at the bottom for a summary. Now we have several terms, so this might take you a couple of pages. So just you can use the first page if you need to, all the way down to the bottom, and then put your summary at the bottom of your second page. The notes, or the terms that you need to put on the left-hand side of your notes, are going to be cell membrane, concentration gradient, equilibrium, passive transport, diffusion, facilitated diffusion, osmosis, isotonic, hypotonic, and hypertonic. And again, I wouldn't go ahead and list these all out now because you don't know how much space you're going to need for each one, but just make sure at the end that you have each of those terms included in your notes and the important information about them on the right. So over here is where you're going to put your notes, here on the right-hand side. And then if you have questions as we're going, they can go underneath the terms over here. That way when you come back in tomorrow, if there's something that you didn't understand or you need to ask a question about, you can remember what that was. All right, the first thing is talking about cell transport. Why are we concerned about cell transport? Well, there are about 100 trillion cells in your body, and those cells all have to move things in and out of the cell to stay alive. So our question is, how do they get these substances in and out of the cell? There's two main categories of ways they can get things in and out of the cell. That's going to be passive transport and active transport. We're going to be looking at passive transport today. We'll focus on active transport tomorrow. All right, so when it moves, the cell moves things in and out of the cell, it's going to have to go through the cell membrane. All cells have a cell membrane. This is what separates them from their outside environment, and it kind of functions as the gatekeeper. It's what controls what goes in and out of the cell. And because it's controlling what goes in and out of the cell, it doesn't let everything in and it doesn't let everything out easily. It's called selectively permeable. Sometimes this is called semi-permeable, but it means that the cell membrane will only let certain materials go in and out. And you can see down here, there's a picture of a cell membrane. It's a bilayer, so it's two layers back to back. It's called a phospholipid bilayer because of what it's made out of. Here's another diagram of a cell membrane. Again, you can see those two layers. And there's things called protein channels that will allow materials to move through the cell membrane, and protein pumps that we'll talk about when we get into active transport. Another function of the cell membrane is that it maintains homeostasis for the cell. And we talked about that when we're talking about living things needing to respond to their internal and external environment. Homeostasis is keeping their internal environment stable. So the cell has to be able to balance things. It has to be able to balance its temperature, how much sugar or glucose it is taking in, how much water it has in it and outside of it. So all of these things have to be kept in balance and maintained through homeostasis. And the way they do that is through active and passive transport, monitoring, controlling what goes in and out of the cell so you have that balance in and out. All right, one of the terms that you're going to be hearing as we talk about passive and active transport is a concentration gradient. So you need to know that a concentration gradient is where there's an unequal distribution of particles across a space, an area, or a cell membrane. So when we're talking about things moving up or down the concentration gradient, moving down the gradient means it's going to be moving from a high concentration of that particular substance to a low concentration for passive transport. And then if it's moving up the concentration gradient, which we'll talk about for active transport, that means it's going from a low concentration to high. So in your body, your blood has a high concentration of carbon dioxide, but your lungs have a low concentration. And so that material is unequally distributed, and it's going to move down the concentration gradient. Another term that you'll need to know is equilibrium. And equilibrium is when the concentration of particles is equal throughout the space. So it's the same on both sides of the cell membrane. When this happens, the movement for diffusion is going to stop, because then it's equaled out, it's even on both sides. There's not going to be any like net movement as one side gains particles and the other side loses particles. There can be random movement back and forth. The particles aren't going to just stop moving. There is going to be some movement, but when they're equal, there's not going to be any net movement. All right, now we're on to passive transport. And passive transport is when a cell does not have to use energy to move particles across the cell membrane. It happens when particles are moving from a high concentration to a low concentration, so they're going down that concentration gradient. And since that happens naturally, it doesn't require the cell to use any energy to move those particles. There's 3 types of passive transport that we're going to talk about. 1 is diffusion, 2 is facilitated diffusion, and 3 is osmosis. All right, so our first one that we're going to talk about is diffusion. And diffusion can happen with many substances. It can happen with a cell, it can happen with gases in a room, it can happen with liquids, as it's showing here, these liquids in the beaker. And diffusion is moving the molecules from a high concentration to a low concentration. The molecules are bouncing and moving around. They have kinetic energy. Kinetic theory says that they're bumping into each other, and when they're bumping into each other, they're going to spread out naturally. Again, this requires no energy on the part of the cell when it happens in a cell, because the particles are moving and spreading out. So you can see here where they've put red food coloring in some water, and at first it's highly concentrated here in the center, low concentration out to the side, but over time, as those particles move around, they spread out until they reach an equilibrium, which is where they're equal throughout, and you have the entire water being colored red instead of just one area of the water. So that's an example of diffusion. Some other examples would be if you are spraying an aerosol or spraying some kind of a perfume, that scent is going to diffuse through the air. When you put sugar or even creamer into coffee or tea and stir that up, that's going to diffuse. It's going to slowly move. You can see here in this little diagram that those particles are slowly diffusing from an area of high concentration to an area of low concentration. And diffusion works well for small molecules with the cell. Molecules like oxygen, water, carbon dioxide are going to move in this type of process in and out of a cell. There's also our second kind of passive transport that we're going to talk about is facilitated diffusion. And facilitated diffusion is where the materials cross the membrane. They still don't have to use energy, but they're going to have some kind of a channel protein or some kind of a carrier protein in order to help them go across the cell membrane. So here's an example of a channel protein. It just gives that specific molecule a channel to go through the cell membrane. And this is going to be used for things like sugars. Larger amino acids can go through these particular channels. Let's see if we can get this to work. So this is going to show us a carrier protein. And a carrier protein, here we go, is going to, again, it's going to help the particular protein move through the cell membrane. All right, our last type of passive transport is osmosis. And osmosis is, there we go, diffusion of water across the cell membrane. Again, it's passive transport, so no energy required. It's going from a higher concentration to a lower concentration. It's the same thing as diffusion of any other materials, it just has a special name. And again, the water is going to move from where there's more water to less water, from a higher concentration of water to a lower concentration of water. So you can see here in the animation that this side has a higher concentration of water. This side is lower because it has sugar dissolved in it, so it's going to move to the lower concentration side. This here in the middle is a selectively permeable membrane. The membrane allows the water to go through, but not the sugar. The sugar can't permeate that membrane. Now, the reason osmosis has its own special name is because it's super important to the cell. It allows them to maintain homeostasis. Most multicelled organisms, unicelled organisms are subject to osmosis because they're surrounded by water solutions. They have mostly water in them, mostly water outside of them, and because they are surrounded by water molecules that are mostly water, osmosis is a really important process for cells. See if we can get this animation to work too. All right, this is going to show that when the concentrations are equal for water, the particles are going to move, have about the same equal concentrations on both sides. But when we add salt to this side, this then has a lower concentration of water. So it's going to pull the water this direction. It's going to move the water to the lower concentration side. Here's what can happen with osmosis, and here's why it's important for cells to maintain that balance of water. Here is a red blood cell, an animal cell, and here's a plant cell. When it's normal, there's an equal concentration of water inside and outside the cell. However, if something changes that causes that concentration to be different, then the cell can actually burst. More water can move inside the cell than is outside. Or the opposite, more water can move outside of the cell and you can end up with a cell that gets shriveled, which is not good either. Now we're going to talk about hypotonic, isotonic, and hypertonic concentrations, the different concentrations for osmosis. If the concentration inside the cell of water is equal to outside the cell, then that's called isotonics, which iso means equal. So you can see here in this picture, both the cell and the outside environment has 10% salt. So since they're equal, they both have 90% water, 90% water. Water's going to go in both directions because this is an isotonic situation. In that case, the cells will not change size. So here's the cell before and here's the cell after. Water's moving equally in both directions. There will be no change in the size or the shape of the cell. If it is, however, hypotonic, hypotonic means that the outside is a lower concentration of the dissolved solute, so it's going to have a higher concentration of water. So here you can see in this diagram that there's 80% water inside the cell, but 90% water outside the cell. Because there's a lower concentration of water inside, the water is going to move into the cell. If the water moves into the cell, then that means it's going to take in water, it's going to start to swell, and it even can actually burst. And again, hypo means less than or below. It means this outside solution has less of the solute than the inside solution. So this is hypotonic. And our last is hypertonic. There we go. Hypertonic means that this is high in dissolved solutes. So you can see the solution outside of the cell is high in dissolved substances. A lot of times that's salt. And because of that, water is going to move out of the cell. Again, these are all passive transport. So that means that it's going to move from high concentration of water to the lower concentration of water. Here we're going 90 to 80. Hyper again means more or above, a higher dissolved solute level. And because the water is moving out of the cell, it can cause it to lose water and shrink. And so the cells will shrivel. And so here's an example of what you might see the cell before and the cell after. Just to review real quickly, here's a picture coming up. Maybe. That shows, let's go back, a hypotonic, low solute outside the cell, isotonic, equal outside the cell, and hypertonic, high solute outside the cell.